Well, welcome everyone. Exciting to kick off this next session. As Tim described it this morning, kind of a new session for ARM on the pioneer's tax, the challenges and opportunities of being at the forefront of innovation. I think as we look across the sector, right, and certainly the sector continues to evolve, I think everybody in this field is really a pioneer. What we're looking at today, we got together a phenomenal group here that's really leading that forefront of it. We'll dive into this a little bit, but as a company leading a new technology that has never been in front of the regulatory bodies before or moving that towards the market, what are some of the challenges that are really faced inside of that? And we'll get to introductions in a second, but excited because as we look across this panel here, what you're going to hear about is we have gene editing, in vivo gene editing represented here. We have cell therapy for new applications represented here, and we also have tissue engineering. So with that, maybe we'll just go around real quick. And if you could introduce yourself, kind of your background, what product really falls into this category for you? And then what stage of development is that in, whether that's an IND stage, whether that's clinical phase, or whether that's commercial? And maybe we'll kick it off with you, Laura.
With me? Oh.
Yes.
So my name is Laura Sepp-Lorenzino. I'm the Chief Scientific Officer at Intellia Therapeutics. So we are developing CRISPR-based medicines. So CRISPR is a technology that allows us to hone into a disease-causing gene and introduce a precise change. So, and we are, we're a clinical stage company. We're developing, there are 3 prongs to our strategy. That's not my phone. One of them is to make sure that you have a, robust tools that allows you to have capabilities to be unlimited in the type of genomic surgery you want to do, right? Because, there are going to be different gene changes that are going to be responsible for disease. And then having the means of delivering this CRISPR machinery in a safe and effective manner. And that we do in vivo, where CRISPR is a medicine. And, for us, we started with diseases, rare diseases in which there is a risk benefit that supports the development of a new modality for medicine. And then we're also using CRISPR as a tool that allows us to engineer cells, and that we're applying for immuno-oncology and autoimmunity. And yes, we are in the clinic for 2 programs that are advancing to late-stage clinical development. And we were the first company that demonstrated clearly that CRISPR works in humans as expected with, the properties that, we designed for.
Thanks, Laura. Johanna?
Johanna Rossell, Chief Commercial Officer for Enzyvant. Enzyvant Therapeutics is a company that works in the rare disease space, and we have a product that was approved by the FDA in October of 2021, and it's called Rethymic. Rethymic is for kids that are born without thymus. So we reconstitute the immune system with Rethymic for these kids that are born with congenital atypia. And Rethymic is a tissue-based, re-engineered tissue-based product that once it's processed approximately between 17 to 21 days, it's implanted in the thigh of the kid, and between 6 months to a year later, the immune system of the kid, of the child, starts working. These children without Rethymic and with standard of care, in general, in a natural history study, they don't survive past 3 years old. So we're actually working on a disease that it's deadly, but at the same time, as of now, we only have treated, kids during a year. So in the US, there are kids still waiting to be treated. So the hope is that with all the plans that we have, as soon as a kid is born with congenital atypia, they should be treated with Rethymic. So our vision is that we change, and congenital atypia is not a deadly disease anymore.
Well, thanks, Johanna. Seth?
Hi, Seth Ettenberg, CEO and president of BlueRock Therapeutics, pluripotent stem cell company. The first program that we're working on is a Parkinson's disease midbrain dopaminergic neuron cell therapy. That is to replace the cells that are lost during the pathogenesis of Parkinson's disease. We're nearing completion of our phase 1, and happy to be here to share some of those lessons. As way of introduction, I was there for the early days of Kymriah, built the cell therapy team, left well before many of those lessons were learned in an autologous cell therapy, and then went on as a employee one of a startup company that was doing similar autologous cell therapy. And I think it's really, it's great to get to a forum where we can share some of those experiences to help those that are pioneers themselves in the field.
That's great. Thanks, Seth. Andrew?
Andrew Bellinger. I'm CSO, CMO at Verve Therapeutics. We're developing in vivo gene editing medicines for cardiovascular disease. So our therapies are lipid nanoparticle delivered targeting the liver using CRISPR technologies, including our lead programs use base editing, which is a second generation variant of the CRISPR process. And we're developing these therapies initially in patients with high-risk genetic subforms of familial hypercholesterolemia, but the plan is ultimately to develop them in broader indications to treat the more common forms of the disease. And our lead program, Verve 101, is in the clinic in the UK and New Zealand with phase 1 readout later this year.
Great. Thanks, Andrew. And I'm Jeff Ross, CEO of Miromatrix, where we are bioengineering organs for transplant. So we're looking at how do you actually solve one of the large unmet needs on the other side, with liver and kidney being our lead programs. I guess before we jump into some of the additional questions, maybe just we can go through real quick, what's the opportunity for the patient and the field with each one of the technologies? Maybe we'll go in the reverse order.
Reverse order, okay.
Serve it up to you first, Andrew.
Well, I think probably the obvious contrast is with a lot of the programs that we've heard about at the conference here, which are primarily rare diseases. I think that the kind of premise and the opportunity at Verve is to tackle actually more common diseases, which probably seems counterintuitive and is actually one of probably the pioneer taxes that we've faced as a concept. But I think the promise that we see, the vision that we see, is that actually, for a lot of these very prevalent and very life-threatening diseases like atherosclerotic cardiovascular disease, the current care model is really not working very well. And you see that, obviously in the burden of disease, which is large, it's the leading cause of death in the world, but also in the individual patient experience, which, for many of these patients, the presenting symptom is a heart attack, and actually in about 50% of those, death. So, we think that one-time therapies are actually very natural fits for conditions that you experience over long durations, and where the treatment needs to be sort of, long in duration, because the pathophysiology of atherosclerotic cardiovascular disease is really your lifetime accumulation of risk. And so, that's the vision, is to, fundamentally change the way we care for, patients in cardiology by treating them a little bit earlier and reducing their lifetime risk of
That's great.
Life-threatening events.
Seth?
Yeah, sure. So the vision at BlueRock is simple in its understanding, but bold in its technical execution. And what I mean by that is there are several diseases in our body, you just described one in heart failure, or others where the pathology is actually the death of a cell type that leads to then the consequences of. And so our mission is simply to identify those cell types, rebuild them from an induced pluripotent stem cell to the near same example that your body has, and then integrate them back into that system. So incredibly technically complex to do, and as of yet not demonstrated well, but simple in the approach of how we go about it. And so Parkinson's disease set up as one of the best examples of that, a very well understood pathology that is due to the loss of a cell. The disease itself, we still don't really understand, so cure still seems like a far reach away. And so our vision is that doctors will have the ability to reach into a freezer with a ready-to-go cell type that then one-time administration alleviates what is an extremely scary diagnosis for our patients. That is that while you'll have a medicine that we can treat you with in the get-go, when you first show up to the clinic, both you and the treating physician know that over the next coming years, for some 5, for some 15, that's still unknowable, you will not respond to those drugs. And so the promise of regenerative medicine is really compelling to bring back that function that your body has lost. And Parkinson's is really an incredible starting point for us there.
Thanks, Seth. Johanna?
So for us, being able to demonstrate the promise of regenerative medicine and being able to treat these kids and change the lives of these families. You know, you have a kid, this is an ultra-rare disease, very happy, newborn screening, the kid has congenital atemia. So we have demonstrated for a year that we can commercialize, so the vision for us is that we can expand. We will talk about the manufacturing difficulties of doing this, and we have invested on a new manufacturing facility that will allow us to expand in the US and outside of the US to transform, like what congenital atemia is, and we're also studying rethymic in other areas. So for us, it's the promise of changing the lives of these families.
Great, thank you. Laura?
Yeah, and I think there are commonalities, right? We're all aiming to change medicine, right? And really go from treating disease to potentially having curative, transformational therapies, right? And these are benefit not just for the patients and the families, but also for the healthcare system, where with early intervention, early diagnosis, but also having technologies that become new therapeutic modalities. And for that, you need to demonstrate, the quality, the consistency on performance, all these quality attributes, right, of your drug and the performance. And that's what's going to allow you to build trust, right? And that's, for, again, these all new technologies, right, whether they were antibodies or oligonucleotides, right, went through these, demonstration period. And I think that this is very important for all of us to ensure that we have the highest quality. At the end of the day, it's going to be, safety, it's going to be efficacy, it's going to be duration of the effect. When we're talking about cures, it's not just, 10 years benefits, right? It's looking at really, enhancing, changing that outcome of that person, ideally for the rest of that person's life. And, and I think we're all privileged to, be working on amazing technologies with, really great teams that allows us to do this. I feel incredibly lucky.
Yeah, thanks, Laura. I think you queued that up well, some of the commonalities that exist up here of kind of pushing the edge or finding that opportunity to think about how do we solve that with looking both at, what's the payer impact? How do we work with the medical professionals? Sometimes that's in a very different realm than what we're doing today. And certainly on the regulatory side as well. So maybe let's dive in, start talking about what are some of the challenges that you've faced over this and the journey in terms of where you're at. And maybe I'll kick it over to you to start with, Seth. Kind of on the regulatory side, I mean, if you look at your therapeutic of taking iPS cells, iPSCs, treating Parkinson with that, what's been your regulatory experience as you're taking that and really moving that to the forefront?
I was thinking of this question as we were coming up here across the different modalities that I've worked, and there's sort of a common theme for me, which is remembering who's sitting on the other side of the table and when they're reading your documents, and appreciating that overworked and oftentimes not enough time to go in depth into your field, and remembering that you really have to take your time in a literature section. You really have to take your time both curating it and explaining it to bring the regulator on board. And many of the, what I would say, difficulties either by clinical hold or by just lack of understanding could be avoided by doing a really diligent job and a really thoughtful job and then testing it with those that aren't expert in the field before you send it in. I'd say to you that most of the conversations we have, especially the early ones, the pre-INDs or the just running up, are incredibly flexibly minded in terms of their approach to you. But then when it comes to approving and especially manufacturing and the characteristics and the analytics that are required to get them there, you really have to bring them back to all of the learnings that have been made often over decades. While you're very new into human beings, there's usually 10 to 20 years of understanding of your field before you get there. And so I would say the one common lesson that is learned over and over, or tax that I would say that I've paid over and over again, has been just not appreciating the burden that you're putting on someone else that hasn't spent the same 20 years in your field.
Yeah, and I think that's helpful. I mean, as you look across this, I think all of us or most of us have been on clinical hold at some stage. Partially, I think, of how you're at the forefront, right? As you kind of talk a little bit about there, is how do you actually educate the regulatory bodies at the same time? Because many times they've never seen this before. And sometimes they don't even know the questions to ask until you're actually submitting the IND or you're putting that in front of them. You know, in our pre-call and in some of the conversations that we've had as a group here, I think, Andrew, you brought up an interesting thing here that I'd love you to talk about as well. And that is, you know, how do you put together a team in some of these areas for things that haven't been done before? Because you can't go out and hire someone who's done this and bring them in. What are the challenges there?
Yeah, I mean, I think they kind of break down into three buckets. One is, you know, inspiring them to believe in your vision, which I actually think is the easy part. You know, because I think there is, you know, at least for us, I think a very clear and simple kind of mission. The second part, though, is that you're often hiring people with skill sets that are not well represented in pharma or biotech. So, you know, in the gene editing space, you know, we're certainly looking at a lot of people with editing backgrounds that, you know, really just don't exist, you know, in the industry for very long. So you're trying to take people who, you know, often have academic backgrounds or, you know, are relatively early in their career and adapt them into an organization. And then the third part is actually organizational, because you often can't build these companies the same way you would a more standard biotech. The kind of team structures need to be different because of those unique skill sets that you need. And the complexity of the drug product. I mean, Laura talked about the quality attributes. You know, there's a lot of quality attributes when you have all of the components that you know, we have in these in vivo gene editing products. And so you really have to put a lot of time and effort into how do you structure the team to, you know, create that knowledge base, the experience, you know, bring in the experience, and also create a culture that, you know, when you don't have a playbook, you have to readjust your plans so frequently that you need that nimbleness that, you know, sometimes is harder to construct in more traditional pharma structures.
And then, Johanna, from your perspective, you know, as the product that's already commercial and in a field that hasn't had a lot of commercial products, tissue engineering, what have been some of the big challenges for you that you'd like to share or to overcome?
So I think the biggest challenge we had was manufacturing. We were very lucky to work with an academic center that was doing the procedure for many years, and that gave us a lot of years of data in North Carolina. However, when we had the first round of changing the manufacturing facility to get approved by the FDA, but we got a CRL. And the CRL was based on, obviously, how do you change an academic center to be able to be a GMP facility? In our case, the donation of the time of the human thymus comes to the institution, is manufactured there, and implanted at the same institution. And we needed to make sure the FDA felt comfortable, to your point, that is, the quality is there, is sustainable, you have all the safety and, you know, no cross-contamination, etc. So what you do as research, even if you're doing a clinical trial in phase 3, is never the same as being a GMP facility. And we need to step back, build, and change the whole chain of manufacturing of rethymic to be able to be approved by the FDA. So,
I guess as we move on to the next question, and I think we'll hit on manufacturing, because I'm sure all of us have some component of manufacturing to talk about, or what some of those challenges look like. You know, what have been some of maybe the lessons that have been learned through this process that could maybe be passed on to those behind it? And, you know, kind of a loaded question in some ways, because as part of the Pioneer Tax, I think you as individuals and companies take a lot of burden on. So some of that is, you know, that front mover and stuff like that. But, you know, what would you be willing to share to kind of pass on some of that knowledge.
Yeah. If I continue
Absolutely.
with what happened to us. So normally for all of us that have been in pharma for a long time, whenever you get a CRL or response from the FDA, you want to go back and have the conversation. But this time we took another approach because we learned by working really close with the FDA at the beginning of the process, we learned that we were teaching them because they have never done a manufacturing of a tissue, re-engineer, regenerative medicine was new for them. And even though we had a very close collaboration with them, when we received the CRL, we can see the questions need answers. Doesn't need to go immediately approach the FDA to brainstorm how do we do this. So we took our time and we did a type A meeting where we came with solutions, with ideas of how to solve the areas where the FDA didn't feel comfortable and in a way that they felt comfortable, but that's something that we could do, that, it was an approach that would make sense, that was sustainable. So that was the approach that we followed, and, almost 2 years later, we got the approval. So working close with the FDA, but also knowing that you are going to be teaching some of the areas that, they don't have the expertise. That's why we are pioneers. Nobody has done it before, so.
Absolutely, absolutely.
If I could just pick up on Johanna's point, because you talk about, it was interesting how you go to the taking your time, because I think often as a pioneer you feel there isn't, we can't take time, either by pressure behind us or pressure of funding and believers in what you're doing, because it's what you're transforming is so unbelievable. And so I think it's a really good lesson you're educating us with, and I wonder if, at least in my own mind, I start to think about the fact that those are dialogues that we do need to be in and often, and it's getting harder, especially inside the U.S. It's getting harder to get time spent with regulators to get that reflection. And so part of our lessons have been the strategies can't always be fast to fail. Some of them we have to take time before we go fast to fail. It's expensive to do it that way, but the dividends, if you believe in your product, the dividends of that extra work on the manufacturing and the analytics and the characterization have always paid off.
Definitely.
But you still need to have enough time to get there, which is right now in many of the companies that I consult with, the young companies, they just don't have the cash to say, let's take our time.
Yeah, and how do you solve that in some of, I think we've all had unique solutions through the funding side, but I think you bring up a great point in there too, Seth, is, being first, as you're raising and doing other things, many times there's a formula or something like that, and usually you don't fit into that formula, right? So to find the necessary capital to really move that forward is, I think, one of the key pioneer taxes, because once you're successful, it's much easier for people behind you to find funding to become a fast follower, or they're utilizing your playbook a little bit. What are some of the ways panel members have solved that, or how did you tackle that? I don't know if anybody just wants to open that up.
Well, I think, as you say, every company is different, and each company kind of chooses its own message to funders. I think differentiation is important, and we had the benefit of Intellia kind of being in front of us and providing at least investors some confidence that there was technical probability for success. But you still have to kind of message the different. We were a very therapeutic area focused company, which 5 years ago cardiovascular was not a particularly hot area for investment. Common diseases was a pretty improbable area for gene editing to be focused on, and so I think you have to both differentiate yourself, but also build conviction in the approach that you're taking. And for us, it came down to the fact that the human genetics was so clear that these genes, PCSK9, ANGPTL3, LPA, are incredibly well-validated targets for intervention, both from the human genetics and also from other pharmacologic methods like siRNA or monoclonal antibodies. And so we could build a case, a scientific argument, that The biological risk was extremely low. The technical risk, was being de-risked by Intellia, and then it really came down to kind of our ability to execute. And so, I think our strategy as a company was to be very focused and execute, and that built, I think, investor confidence.
That's great. As we move into a little bit and we kind of look at, this was brought up as well, the regulatory side. All of us need to make it through that aspect, right? And we've heard a lot at this conference on the European regulatory. We heard about the FDA. Would love to get some of your guys' perspective on what has gone well or hasn't gone well from the regulatory side. And maybe I'll pass it off to you, Laura, to kind of talk about your guys' experience on that side.
Well, we had a very positive interaction with regulators worldwide. We have 2 in vivo trials in multiple geographies. We just got clearance to start our phase 2 in the U.S. And it was a very smooth process. So, I think that what we've done is taken advantage of all the opportunities to engage regulators, right? Pay really close attention to what they're asking you to do, and really, try your best to not only do that, but, push the envelope, right? To answer all the questions, fully and clearly. And, that has served us really well. This would be our 3rd IND approved in the US. The other 2 were for ex vivo programs. So, you build on the knowledge, right, from one and the others. The other thing that we've seen is that across, multiple, regulatory agencies, it's not very different what they're asking for, right? There are some nuances, but, in general, what they're asking is the same. So, it's important that your package at the start is complete, and addresses their questions. It helps. Andrew was talking about, getting a team, and I think that's really important. At Intellia, we are the veterans, right? But we have, a bunch of us have had multiple approvals of important drugs, right? In using different modalities, from small molecules, antibodies, cell therapies. So there is bringing all that experience together helps inform how to approach some of these challenges.
Yeah. Have you noticed a difference in the EU versus US, or any strategies or differences on the regulatory side there?
Yeah, and on all of them. Look, it's paramount for regulatory agencies to ensure safety, right? So that is for everybody. In the US, for example, the approach has been a bit more linear and slowed down compared to other, and I'm talking about dosing patients, right? So, sentinel patient, wait some time, follow. In other geographies, that is different, but in general, for example, for gene editing is your precision and specificity, your on target and off target, they all care about the same, right? Making sure that, you don't have off tissue edits that would be deleterious. They all care about that. The question of germline editing, right? So are you unintended, having edits that can be passed to progeny? Again, they all care about that. So there are no fundamental differences.
Oh, great. Seth, how about,
you
on the cell therapy side?
So I think I agree with the fundamental questions are the same. Mm-hmm. I'm not sure the approaches to me in cell therapy have been the same. At least, and it's a bit of forecasting now for the European pieces, at least for the PSCs right now have been focused in the U.S., is that the bar for the analytics and the quality within the European agencies, specifically the EMA, has started to go up quite a bit, which creates a new burden on especially young, innovative companies to be able to meet that burden. Now, we just talked about spending the time in the manufacturing, so I think it makes sense. I'm not sure we're doing patients a safety benefit, and so I think we as users need to make sure we're working with the EMA to watch out for the loss of innovation that may happen here from that bar going up. It's not an easy spot and an easy line to call out. So, but your comment on I agree with the dosing piece in the U.S. That we seem to still be stymied by more traditional approaches there than we've gotten away with in other jurisdictions. But the quality piece, I think, is a burden that we have to deal with. Otherwise, things, especially in the next couple of years, are just not going to come through.
Yeah, and particularly on regenerative medicines, right, where you're aiming that your cell will be there forever, right?
Yeah.
So I can imagine that ensuring that the quality of that,
Yeah, exactly.
should be very high. But look, like everything, I think that, as a field, we're going to be building that, evidence, right, of what works and what's safe and what's efficacious and what's durable that, will help everybody. So
we talk about this as a pioneer's tax, but there's also a pioneer's burden of not spoiling the field for others, being thoughtful and
that
careful.
too.
And something
Be
we all
careful.
always worry about is that someone will do something that then often is translated into the entire field set.
Worries me like biohackers and all this weird stuff, right? Like, don't do it. Don't do it.
And I think that comes back to a little bit of, that need to go fast, but not too fast, right? There is a certain balance inside there of that urgency, but don't create a challenge for others behind you as well. Johanna, as the, having a commercial product already through approval in the U.S., any advice or thoughts or learnings from that of what went well beyond what you already kind of spoke about?
So to build on what Laura was saying, also take advantage of the designations. The RMAT, the breakthrough designation, the orphan designations, they all come with advantages of opportunities to talk to the regulators. I think, like we always check, but what comes with it. So I would say that. And in general, not only with the regulators, but I would say our experience in the US commercializing Rethymic bring us a window to the future of gene and cell therapy. Remember I told you that our academic center provide us, and we work with them to make sense of data for 25 years. So with that came a very strong value proposition. And we have been extremely surprised with the receptivity of the key stakeholders, payers. Most of our patients are in Medicaid in the US, because maybe these families didn't start being in Medicaid, but because these kids have so many issues, they ended up, or the family, or just a kid being in Medicaid. And they have so many issues from the healthcare system, hospitalizations, et cetera. But also, if you have a kid with congenital atemia, even before COVID, the whole family needs to isolate because this kid has no immune system. So the other kids need to be homeschooled. One parent needs to work and be able to come back and not bring infections. And not everybody can work in, Zoom and jobs like that. So they ended up in Medicaid, and most of our patients, a big percentage are in Medicaid, and we have been surprised when we show our value proposition, what Rethymic brings. We have had very good receptivity, and I feel like this is not like something that I can tell a new drug, because it's not going to have 25 years, and maybe 25 years is the most robust data you can have. But what is true is that the promise of having the outcome data and the promise to not try to extrapolate or surrogate data, but coming to the to payers and everybody that will support your drug with a plan. Like, maybe today I don't need to do outcome-based because I have the data. Maybe today you need to go through outcome-based, but in the future, there is a future for these drugs that are obviously more expensive because it's a very complex manufacturing. But if you are able to demonstrate that, this is the right thing for the healthcare system, for the family, and for the payer, there is success. So,
well, that's great. As we think about this, maybe one more question, then we'll open it up, see if there's any questions before we go a little bit further. But throughout this, what surprises have you encountered?
Well, I think we talked a little bit about manufacturing and some of the challenges, and I think yesterday at the session on manufacturing, we heard about how a lot of AAV companies have been moving away from internal manufacturing. I think in the gene editing space, it's a little bit more complicated, and most companies seem to be taking up more of a hybrid approach. And that's really driven by the fact that, when you're developing these new modalities, like gene editing, you have new quality constraints that come up. In vivo applications are more challenging than ex vivo. The quality constraints are higher. Guide RNA, purity is a challenge in the field for the manufacturing space. So I think it's pushed a lot of companies to kind of have to be a little bit more creative and coming up with solutions that leverage both internal expertise as well as the external CDMO capacity, which in principle is growing pretty rapidly, in part because of the COVID vaccine work in LNP and mRNA. But still is probably not where it's going to need to be, as we're thinking about phase 2, phase 3, pivotal trials and commercial space. I don't think the field is collectively really ready for that, for in vivo at least.
I think we've built a system over many, many years that is very good at piecing apart incremental changes. The kinds of late-stage development and the expectations of payers and the system that we've built can pull apart and tease apart small differences very, very well. What has surprised me is that we're flexible in conversation, but not in practice yet, around the differences of really transformative changes, of what we do from a payer perspective, what we do from a even treating physician perspective, of how to create that system that allows for the doc to treat a patient that's been one time treated. The remuneration there, so we're flexible in conversation, but even in the earlier sessions today, we seem less capable of executing while the science is moving incredibly fast. Now we have multiple validation points across gene editing as well as cell therapy of products coming to market. In Europe, we have some products coming off market. Mm-hmm. We don't seem yet to be able to how to translate and execute on that transformation. And I think for me, that's the surprising piece yet. The conversation's there, but we don't yet have the practice of both from the physician to the patient to the physician to the payer system down, right?
Have you guys started doing some of that stuff to try to get in front of it?
Never early enough.
Yeah.
But certainly they're active on our minds and active on. So we're. I didn't describe it. BlueRock is a wholly owned subsidiary of Bayer Pharmaceuticals, and we're lucky in the sense that that's a 150-year-old company with lots and lots of experience there. And now looking at us as an innovation engine also means that we need to innovate in that space as well. And so yes, we're having innovation conversations there, but the practice, if you look in front of us, of the pioneers in front of us, they haven't been able to put into practice really novel methods there yet.
Other thoughts?
Surprising. I think the underestimation of how complex the whole process is. Like not only the manufacturing, but also the journey of the families that need to, get the medicine. In our case, as I said, we are only in one institution, and that means people from faraway states need to go to North Carolina to do the treatment. And how is like everybody needs to get involved. How are you going to move this family to come and have the operation and then ensure that they are healthy enough to go back, and how obviously the government, if it's a payer that is Medicaid or a commercial payer, it doesn't end on just you get the treatment because afterwards, I think I mentioned, it takes like between 6 months to a year. So first you have been in a waiting list, then you get the treatment, and you can't get too happy because you can, do the things that you think you will do. Once you get an operation, you need to make sure referring physicians and institution and everybody is behind the patient until it's determined that the immune system is working. So for each of these patients, there is a huge, follow-up And the fact that we have a hub called Enzyvant Connect. I have been in many hubs, in different pharmaceutical companies, but this is the most involved that I have seen, and really follows from the possibility of getting the treatment to the moment that your kid is 3 years old and can do normal things. So.
And I think that was talked a little bit about this morning, right, on the patient advocacy and stuff like that, of really bringing that back forward, because I think as many developers, sometimes we can lose sight of what that patient journey is, right? Or we're hearing it more from the medical professionals. And to get close to the patients and really understand that, I know for us has been really important. And I think that's a great lesson learned too, is get close to the patients early to understand what that journey and challenges are.
Yeah.
Laura, anything you want to add?
Yeah, so for me on surprises, or, and I'll talk about positive things. One is the speed of innovation. I mean, for example, CRISPR was, first publication, was in 2012. In 2021, we already had proof of concept in humans. I mean, it's an enormous, speed of bringing. So what, now we need to do is, translate that into real solutions for patients, and go in, the discussion we've been having, right? Not only to, the rare disease where you can start, to the large prevalent indications, but also to the N of ones, right? So how, as a field, we're going to be able to have these, access. So, those patients that have these terrible journeys, to understand where they are, right? We do have tools that we should be able to deploy to help them. And that's going to take industry, regulatory agencies, payers will need to find a way of doing, that sector of patients. ARM, others. How are we going to do that to really ensure access worldwide, to these new amazing technologies?
Absolutely. Maybe we'll pause for a second, see if there's any questions in the audience. There's got to be at least one burning question out there.
I cannot see anything, so
I don't think any hands are up yet. So maybe we'll keep moving a little bit. Maybe talk about what are some of the frustrations that were faced, and how do you see the path forward through that? What are some of the questions that you're kind of faced early on? Maybe Seth, we'll kick off with you.
I think some of the frustrations along the journey will have to be on back to pace. Is that multiple times you've heard everyone up here talk about the complexity of what's happening and some of the twists and turns that you go on, that especially as you move into the GMP facilities, that you find out that putting that very intricate work into practice in such a facility is very difficult. And I think patience and expectation of those would really help, because I think I would have tell you that my lack of patience or my frustrations would come from those moments where you had thought you would be in the clinic or you thought you'd be at the next dose, but you're not. And you have now a finding that really says there's quite a bit of work to do here.
Andrew?
It's not a frustration exactly, but I think it's central to the mission for us, which is tackling both these rare initial indications, but also having a plan towards the broader indications, and that raises interesting challenges in terms of clinical trial design. When we're kind of a hybrid in a way. I mean, we are a gene editing medicine, which entails all of the complexity and all of the challenges, but we also are going into more common conditions where there are precedented approaches that have large data sets. And so that creates interesting kind of tension in terms of how you're going to develop the clinical data in larger patient populations as you go into development. So we're still early in phase 1, but certainly we're putting a lot of effort into what would larger pivotal studies look like for something that is both a gene editing medicine, which would say, small trials, and something that, is a cardiovascular therapy for even heterozygous FH, which is our lead indication for Verve 101. There's 1.3 million people in the United States with that. And so, the pivotal trials that have been run historically are about 600 patients which would be the largest gene editing trial, by far, and so those are the kinds of things that I think we're spending a lot of time thinking about, because it trickles down to your manufacturing, it trickles down to everything.
Jen?
A word, manufacturing. So I explained that our product comes from a thymus that is donated, and it needs to be from a child that is 9 months or younger that is having a heart surgery. When the kids are really small, the thymus is on top of the heart, and they can't do any surgery in the heart if they don't remove the thymus partially or completely. And we get that thymus, and that's the thymus that is re-engineered. And first of all, like the donations, even if there are a lot of heart surgeries with kids 9 months or younger, these thymus need to go through tons of testing, obviously to ensure that it's safe for the family, and not only the thymus, but the family, and that there are no diseases, etc. So that whole process is complicated, and then obviously we need to process it and ensure that it has the size of the thymus matches with the body surface of a patient, and we bring that specific kid, or to have the operation. So it's very complex, and that's why now we are doing our new manufacturing facility. Will allow us to have the operation in different centers, not only in North Carolina, and we will be able to get thymuses from different hospitals. So, it's just...
I feel better. I should hang out with you more. I feel better already.
Yes.
Laura, anything you want to add to that, or?
What's frustrating, equity markets. Have you guys seen how red it is? But, on the other hand, it, makes you more, disciplined about, picking what you're working on and what you're not working on. And I think this is, it's a reminder to all of us, right, that, this is something you need to do all the time.
Maybe real quick, Johanna, before we walk into some of the closing things and see if there's any additional questions. As a product that's already out there today, how was that working through with the payers and stuff like that in terms of, did you develop those relationships early on? Did you wait until approval for some of that? Anything you're willing to share on that?
So the education about the disease, definitely across different stakeholders, because nobody knows what congenital atypia is. But, being an ultra-rare disease, they're like, oh, great, until they have the patient. Like, I always say, payers are going to pay unless they need to actually pay. So because, my team will be, no, we had the conversation, everybody, understand our value proposition, but the reality comes when they actually have a patient. And that's when they get serious about learning, the actual disease and what's the value proposition that you offer. So as much as we want to educate, because it's ultra rare, that education in general is good to have. And many of them have created policies for congenital atypia, because there is a drug, and they acknowledge and have already the knowledge. But it comes when the actual family is there. And today is easier because these families have had issues, and this kid is maybe 2 or 3 years old, and they have seen the cost. They can see it in their, in their history. In the future, when we are able to treat this kid as soon as they are born, 6 months, 9 months old, they won't be so costly to the system, because that's the whole point, that, that this kid's not so sick, that perhaps cannot be treated or is already costing. So we make sure now that we have all this data and we collect the information to ensure that, remember, in the past, this was a costly kid. today, it's better to treat them as soon as they are born, and put them in the list and move forward with the treatment.
Thank you. So I think we've all shared, the panel certainly shared, some of the challenges, some of the things that's successful, some of the things that excite you, what's the positive on this as well. I think you all share a common theme of moving into an area that hasn't been there before. You get a lot of barriers and stuff like that, right? And you heard some of them today, but you're all still here. You're all still pushing it forward. Love kind of just final thoughts on, obviously you're here because you're excited about it. You're excited about the future, but would love that perspective of, what excites you about the future in this? And maybe I'll start with Laura and then work back this way.
Yeah, I think we are privileged to be able to work on the forefront of technology that's going to revolutionize medicine. And sounds like, a Twitter tag, but it's for real. We're touching lives, and hopefully we're going to change them potentially with a one and done, and really have someone forget that they ever had a disease and be productive, happy members of society. And and again, it's this amazing responsibility, but I cannot think of a better job, an opportunity. And, the future of medicine is going to be this, and it's going to happen, so.
Thanks. Johanna?
So, of course, being able to help these families and patients, I don't need to motivate my team too much. They know every day is like a family that has a potential of having a better future. So I'm excited to see what we can do. It's complex issues, but it could be done. So, for Enzyvant and congenital atamia, but in general for the whole regenerative cell and gene therapies, that's a whole new future for the patients. So.
I love the word that you used, privilege. I lost my brother to leukemia when I was 10. He was 13. I sat at Novartis and heard the very first stories of Emily Whitehead come back for the very same leukemia that killed my brother, and realized at that moment that this is exactly what we need to be doing. It just needed one patient to convince you that the biology was right. And I'm excited and privileged to be in the field to now do that at a scale in Parkinson's disease, where we're an aging population, and the fear of neurodegeneration lives within every adult that we come around. And to be able to change that path, to be a part of changing that path of what it means for so many, there's no luckier job in the world.
I think we all actually have, pretty much everybody in this room probably has some connection to coronary disease. For me, it's, several members in my family have had coronary disease, died of it. So, I think that the fact, I'm a cardiologist in my clinical life, and when I think about, kind of the early vision of Verve, and what drew me to it was thinking about 20 years from now, 30 years from now. And I'm convinced that this is how we will treat these diseases in 20 years and 30 years. It will be molecular surgery. It'll be the same way patients come in for permanent, irreversible therapies right now for their coronary disease. That's what we'll be doing in the future. We'll just be doing it as an intervention to, lengthen their life and treat their disease in a more, precise, and durable way. And so once you have that vision, that long-term vision of 20 years, it makes it very easy to kind of tackle all the challenges that come along the way. And I agree with Laura. It is a privilege to get to, be on that journey.
Mm-hmm. Yeah, for us, well stated with the privilege and stuff like that. For us on our side as well, as we look at organ transplant, liver patients today, either you're fortunate to get a liver or you don't have that opportunity. And certainly on the renal side, we look at 500,000 patients who are on dialysis today with a 5-year 50% mortality rate. But it even goes a step further of, even when you talk to transplant recipients who've gotten that gift of life, they're also wondering, where am I going to get the next one if they're young enough? In 10 or 15 years, where is that next organ coming from? So I think there's a common theme among everyone up here of, the excitement about the future is that gift of life. How do you create that therapy that now is no longer just treating, but you start to move to a curative front? So, got a couple minutes here. Any questions? There we go. Mike.
Mike. Turn on.
Thank you. Yeah, I'm interested to hear your perspectives on the role of information sharing in advancing innovation. And as the leader, as the pioneer, you're the first one to have the knowledge, which is potentially a competitive advantage. How do you justify that?
So I can answer that. We published a very extensive, paper in the New England Journal of Medicine in 2012. And there we discuss, how we selected guide RNAs, how we demonstrated specificity. Go read the supplement, it's really long. It's very detailed, and we knew up front that, we were going to be helping others that were trying to do the same and that they're pursuing the same. But, we felt that, we had the opportunity, as a leader in the field, to also set the standard, right, of what needs to be done from our point of view, right? What's the criteria, the quality that needs to be documented. And that was, a strategic choice, and I think it's important for the patients as well, right? And the doctors and the providers to really read in detail, what are you doing to bring this new technology to patients? The proof is in the pudding, and you can go read it, Gilmore et al., New England Journal of Medicine, 2021.
I mean, greed has no place in this business. There are so many patients in need when you get to it. To think that you will be the only person or company in that field seems like a fool's errand. And so while you can do both, you can serve both your competitive edge and share, and science will benefit, and teams will benefit, and patients will benefit. And I don't think it's that hard to split the difference.
I think it would be short-sighted to do otherwise, because the regulatory standards are constantly evolving and moving. So I think even if you did believe that you had a brief competitive advantage, in the big picture, in the long view, transparency and collaboration is the right way to go, especially when you're at the beginning of a field, because things are going to evolve and change, and you want to be part of that.
Okay. One more.
Yeah.
Hi there. Question maybe more on the regenerative medicine side. Dr. Ettenberg, maybe most for you. How did you go about on the strategy side, thinking of beginning to kind of unwind the Gordian knot of strategy, where you had this complex cell product going into a disease that is constantly progressing downward? How did you think about patient stratification, endpoint design, and how did you integrate KOL input where you're bringing to bear a new product to this market, and again, a disease that is highly variable in its penetrance and variable outcomes?
There's a lot there. So I'm not going to answer it all, but what I would say to you is, one, learning by committee in all of those things that you said was really important to us. I'll go back to my first statement, which was the simplicity of the hypothesis. In this disease, these cells are gone, and they disappear more and more over time until death. And replacing that, for us, became a numbers game, not a patient stratification understanding. We're too early on to know what that's going to mean long term, but it was just following the biology and the simplicity of the biology. Integration and long-term benefit and all of those questions that you're going to be asking about to which patients, that journey is, we're afoot of it right now.
Well, I'd like to thank all of you for coming up here today, as well as for what you're doing for patients, what you're doing for medicine, what you're doing for the field. Thank you for coming forward, sharing your story. So,
Great,
please join me in thanking them.